Okay, I'm getting to the end. There's 4 or 5 slides now from Graham Simmons, and I think this gets us to one of the more interesting features of the meeting. And as I said in the beginning, when we have 2 researchers, one who finds 25% prevalence in 100 samples, and one who says, I can't find it and I've looked at 800 samples, there's something strange there. And I'm going to show you here the assays that individual researchers have been working with are almost identical in their sensitivity. So what's going wrong? So Graham presented the REDS study. This study is in 4 phases. The analytical panels, pilot clinical studies, clinical sensitivity panel, and the blood donor clinical panel. I'm only going to talk about phase 1 and phase 2 because this is just finished. This is, I think this phase has been completed, but the results haven't been released yet. So in effect, I'm only going to talk about the analytical panels. So the analytical panel at the moment was determining the sensitivity of various assays, nucleic acid detection methods for XMRV infection. The idea was to compare the limit of detection accuracy. They used a whole blood panel spiked with XMRV positive cells, or a plasma panel spiked with supernatant containing XMRV. And now you see the center of the controversy. Okay, here is the code here. Here are the groups, the CDC, Graham's group, the FDA, 2 at the FDA, the Whitmore-Peterson Institute, and the NCI. Here is about the cutoff. You'll see that all assays are almost equivalent in their sensitivity. This is looking at whole blood. If I move on to plasma, again, we're looking at RNA copies. You'll see that almost every lab, every assay is equal sensitivity. And so I think we can say from that that the disparate results that we're getting are not due to the sensitivity of the assay. Again, this is basically a PCR-based assay. PCR has been around for a couple of decades. These are highly sensitive assays. People are using different primers to different regions of the viral genome. Everyone is coming up with equivalent levels of sensitivity. And so the different results we're getting are not from this study due to assay sensitivity. And this is where we get into the phase 2 study. The use of whole blood versus PBMCs versus plasma. The Lombardi studies were performed on isolated PBMC and plasma. Large-scale studies for prevalence are facilitated by using whole blood or plasma. And donor, recipient, and other repositories mainly comprise frozen whole blood and or plasma. Now we come to the important point, which is the time of processing of the sample. And what is going to happen here is that once the samples are collected, they will be, well, here we see actually that from the time of collection to the time of analysis can be 2 to 4 days. Now, what can happen in these 2 to 4 days? This is now accepted amongst the researchers of the meeting that something very important could happen in these 2 to 4 days. It could be that the cells lyse. And this is a very important point down here. Studies with cell-associated viruses, that the level of viral nucleic acid in whole blood and plasma can vary with time. And so we've not given, this was not considered at the early days of the study, how long, when we collect the sample, how long do we leave the sample before we analyze it? At what temperature do we store the sample before we analyze it? And what is happening to the sample from the time between the time of collection and the time of analysis? And this is what the REDS 2 study is designed to do now, is to collect the samples, store them at room temperature, store them at 4 degrees, store them for different numbers of days, and try to understand if we have a cell-associated virus, are we getting a low value on day 1? If we leave it around for a couple of days, the cells rupture, and the cell-associated virus is released. I think that's what I just summarized in this slide. So moving forward, where are we going with this? Those of you who followed the meeting will know that the NIH Director, Francis Collins, announced an initiative between NIAID, who have brought in Ian Litkin from Columbia University. There is a CFS study currently underway where they will be looking at 100 CFS positive, 100 CFS negative from 4 geographically distinct regions. At the NCI, we've already taken up contact with Ian Litkin to offer the reagents we have, whatever they would like from us. Our goal has not been to make this an NCI-specific project. From July of last year, we've been in contact with NIAID. Our reagents, our technologies are available. The other thing we thought we would like to do is develop what we've called, this was the first time I got a chance to give this a descriptor, an XMRV protocol website. And what we want to do is encourage researchers to deposit their PCR protocols on this website in the raw form, not to take a look at it and make the changes and say, this sounds nicer, this looks nicer, but deposit the methods in the raw form on a website that can be publicly accessed. The hope is that if there are small points, as many of you know when you're writing a protocol, there are small points that you've adhered to for 20 years and somehow they never make it to the final method. The hope is that if we can encourage people to deposit their methodologies on a website, that will encourage, that this will hopefully point out some discrepancies in the methods, and we hope it will encourage interaction between researchers. As I said, we're split right down the middle now. It's about time we cross the line to find out why we have results that are very positive, results that are equally negative. So this is one thing that we're doing at the NCI. We're just in the process of establishing this open website for depositing protocols. The other thing that we're doing, I alluded to this earlier, the NCI is in the process of it. This is a prospective study, what we call the 100 Cancer panel. This is done in collaboration between the HIV drug resistance program and the urological oncology branch. 100 samples are being collected, and these are being examined by the multiple assays that we've developed at the NCI. And in July, when I gave this talk at BPAC, our goal is to see when things are positive, are they positive in all of these assays? If they're positive in some assays and not positive in others, then I joked about this. We'll put the researchers in the room and let them out when we understand why there's discrepancies. But I think it's important that as we develop multiple assays, that we have some sort of harmony in our assays before we pass those on. So this is ongoing at the moment at the NCI. And I just wanted to finish with one slide. This is actually HIV, but it could be any other retrovirus. And this is to show the life cycle of HIV, the life cycle of XMRV is very close to this. And just to point out that in addition to the work that we've been undertaking at the NCI in terms of developing the diagnostic reagents, there has been an active program looking at the various parts of the XMRV life cycle with a view to potential antiretrovirals. And as I said, here we have the point of HIV infection. There are programs at the NCI where small molecules have been identified that can very importantly affect XMRV infection. Once the virus gets in, here we have the viral RNA that's converted to DNA. There have been studies for one group on inhibitors of DNA synthesis. My own group is involved in another activity of this enzyme, and we're just publishing a paper on it. Once the virus gets in, this is where the integrase worked, and I told you there are already some publications on integrase inhibitors. The virus, we then reverse the life cycle, the viral RNA comes out. There are studies looking at how XMRV RNA is transported from the nucleus to the cytoplasm. The viral proteins are then matured through the action of XMRV protease. The crystal structure of XMRV protease was reported at the meeting and will be in press very soon in Nature Structural Molecular Biology. And once the virus gets out, there are also several host factors involved in these steps, and many of those are under investigation also within several groups at the NCI. So with that, I will close, and I think we'll move on to a break. Yeah. Almost. Thank you.